My topic is research on poultry vaccines, and just can't see. Okay. Okay. I don't know why it keeps going up and down. Okay. First of all, I'd just like to start with what does one understand by vaccine, and it really depends on who you're asking. So. The functional definition of vaccine depends on how it relates and how it fits. Like currently for COVID-19, this is everyone has really their hopes up with the vaccine development. So it's like everyone knows about vaccines, so and it's a way to protect everyone and to get things started and rolling. Similarly, even within the veterinary faculty, the definition or its functional definition is quite varied, I would say. So, for example, say for a farm manager, it is a tool to minimize production losses and meat quality losses. For an epidemiologist, it is a tool to control and prevent a spread of outbreak into new areas and how it would be controlled in current areas. For a field veterinarian, it is a tool to maintain health of the birds and animals and to keep the farm disease free. But for a veterinarian in the lab, it would again change. So if is an immunologist, he's more interested in when he talks about vaccine. Anyone talks about vaccine, he's more interested in what kind of immune response is elicited by the vaccine and by the disease and how it can protect. Virologist is more interested in how the biology of the virus can be exploited in order to develop a more effective, safe, or multi-targeted vaccine. And for a vaccinologist, which where I fit in or try to fit in, is figure out the best way to deliver an immunogen using skill sets of virology, immunology, and whatever new technology is around. Quite a lot of new technology was described by Kurkure sir. So one has to be aware of all those things that are going on in biological research and how it can apply to reach the goals of the farm manager, the epidemiologist, and the field veterinarian, and all this in a very cost-effective way. So that is how the definition varies with different faculty within veterinary. And so today's talk, I would, I'm guessing it's a very broad audience, I would touch on the functional areas as discussed, and we'll divide the talk into two major parts, the disease management and current issues. I won't go too much in detail, and related to vaccines, and also the new developments and the promising new technologies that can overcome these issues. So the disease scenario are there are epizootic outbreaks, seasonal outbreaks is what we can divide it into, and potential threats, new coming threats. So more epizootic outbreaks would be something that are quite there, and every now and then we get cases. So like in bronchitis, infectious bursal disease, chicken astrovirus, IBH, Marek's disease, avian influenza, and seasonal outbreaks which occur during a particular time period of the year, like chicken anemia virus, which occurs more in the hot and humid uh, uh, time of the year. Infectious bursal disease around same time, more like rainy season. So right now there have been quite a few outbreaks of IBD, and there are then these potential threats such as metapneumovirus, parvovirus, malabsorption syndrome causing viruses, and other new viruses. So I'll briefly touch upon these later on. Okay, so uh, within, I don't know why there are lines coming on. Anyway, so there are new vaccines for new diseases. within poultry vaccine research, and such like chicken astrovirus and nephropathogenic IB. Uh, and we can further divide it into new technology for old vaccines such as recombinant vaccines, subunit DNA vaccines, and nanoparticle based vaccines. And also there are okay, yeah, new concepts of for old vaccines like so the same virus, same vaccine virus or same antigen can be used, but presented in a novel or a more effective form, such as immune complex or clone vaccines, and improvement of old vaccines using newer adjuvants and newer vaccine delivery systems, such as nanoparticles and encapsulated vaccines. So the number one step of poultry vaccine research or vaccine research per se is disease diagnosis. And I'm very glad Dr. Kurkure's sir's lecture was ahead of me. So the most part of it is taken care of, and I cannot insist more on how important that is, because unless until a new pathogen is there, or if the diagnostic tools are not there, vaccine research cannot function in any way. So, because you need to have an antigen to make a vaccine, and the antigen comes from the pathogen. So, unless until you can diagnose the pathogens, you cannot go anywhere. So, I won't go too much in detail as it's already been explained, and it's not much different from what Dr. Tracer explained. So, at field level diagnosis, it depends on clinical history, the vaccination schedule, flock vaccination, parent vaccination, and so these especially in poultry, the vaccination and Dr. Kurkre described about the importance of ELISA and serological tests. So serological test, ELISA especially, is very important in disease diagnosis for poultry, because depending on that, you can because there's always poultry are vaccinated heavily for different vaccines. So it becomes difficult at times to differentiate between the immune response, the antibody response that you're seeing, the serological response, whether it is for to the vaccine or it is towards the challenge virus in the field. So looking at the flock immunity, it is possible to uh, uh, I'm live, right? You can hear me, right?
Yes, sir. Yes, sir. We can hear.
Yeah. Okay. It just showed bad internet, so I just wanted to make sure.
It's okay.
Based on the level of response in the flock, one can differentiate between whether it is vaccine driven immune response or it is a challenge virus immune response. And so that is why it's a very good tool for that purpose. And also the age old pathognomic lesions are good tools, and the molecular tools. And rightly, Dr. Putresar explained the importance of molecular tools. So PCR, I don't think we can do any diagnostic work without PCR these days, and sequencing, which is again really helpful tool to understand how the virus is behaving in the field, to study the field viruses, and how there is any deviation from the current field viruses, how they are mutating or evolving. So to keep track of that, sequencing plays a really important role, and of course, histopathologic lesions are also very important. So, so there are vaccine failures every once in a while, and what are the major reasons for it? And these vaccine failures are the driving force for vaccine research, because where those help in identifying the critical control point, where we can make the improvement to make better vaccine. So there are managemental practices are very important. So farms where the management practices are not good, vaccines fail, and it's not because of the vaccine, but it's just the way the vaccine was handled and the animals were handled in that case. Like the animals are in stress for different reasons: overcrowding, heat, high humidity, bacterial load. So vaccines will not work as well. Then there is interference of maternal antibody. This is specifically very important for infectious bursal disease. And immunosuppression. The immunosuppressive diseases, if the immunosuppressive diseases, then you might have the best vaccine, but it will still not do any good. Then there are vaccine and challenge virus mismatch. So as I mentioned earlier, the challenge virus can keeps evolving, and with to catch up, the vaccine has to be changed or to be upgraded to what is in circulation in the field. So, and that is an important reason for failure of vaccine, and as I mentioned, a driving force for vaccine research. So we always have to be on our toes about what challenge viruses are out there in the environment, and whether the vaccines, the current vaccines, are able to protect them and how well they are able to protect them. Vaccine modification, this I'll discuss in a bit, and unique disease scenarios and syndromes. So one interesting concept Poultry mentioned was dysbacteriosis. So here it's something similar to that, where the it's not just one pathogen that you're dealing with. When you're dealing with farm animals and in poultry, there are multiple, not just you also have your good bacteria, bad bacteria, but also there are viruses which are maybe they're not harmful in a certain scenario, but when condition changes, when there are more stress related issues, then the non-pathogenic viruses or bacteria can become pathogenic, and that getting complicated with more other complicating disease agents just shows a very different and very surprising pictures. So one has to take that into consideration while designing vaccines and during diagnosis. And also there are new viruses which keep coming, like I mentioned, like chicken astrovirus was one really interesting case during 2010, around that time. So where we had never experienced anything like that, and even worldwide, there were chicken astroviruses, but the way the disease presentation was very different in India compared to other countries. So that is why it was very difficult to nail it down. But through lot of concerted efforts from our diagnostic unit and our vaccine research team, we were able to figure that out and come up with our own vaccine. So, and successfully were able to control it during those years. And that is a very typical case. And as we all understand, like, in our exams, there's always this out of syllabus question that we are always worried about. So this is a similar thing in nature also. There are always these out of syllabus viruses that come into the farm, and we are just clueless, and then we just have to figure it out. And but with the new tools for research and diagnosis, we are able to figure it out. And so identifying new pathogen is a very important thing. And with globalization, with a lot of exchange of people and animals, animal exchange is not as much as it used to happen earlier. And but we always have to keep an eye on that. And so serological tests again play a very important role. Say, for example, let's take avian metapneumovirus as an example. It's a very important places in Europe. In India, it's not a major issue, at least so far. There are a lot of quite a few research articles coming from Indian universities where they've shown that avian metapneumovirus was present based on antibody. So using serological test, they were able to get antibodies against metapneumovirus. So that indicates that the virus is probably present, or there is something that is cross-reacting with avian metapneumovirus. But we have not really seen a major problem with this particular virus in India, or I'm not sure with the recent research, but very recent research, but till last whatever I have checked, I couldn't find where they've isolated avian metapneumovirus from in Indian conditions. But these are the things that we need to keep an eye because not today, but say tomorrow or 6 months from now, it might be an issue and we might have to take care of it. So it is always better that we are on top of things. So the reason for vaccine failures are, as I mentioned, management issues. So in that, administration of vaccine, the training of the personnel is very important, especially for these like Marek's disease or killed vaccines, which need to be given their subcut. And if you miss a particular few birds, you are putting them at risk, especially if you're giving a kill vaccine, because kill vaccines, they do not transmit from one bird to another, so it's all or none. So if the bird gets skipped, that bird will not be protected. So vaccine administration is very important. The right technique is also important. uh, and poor distribution of mass application methods, especially for ND vaccines. All these vaccines which are given through drinking water, they need to be given in the right way. There are a lot of conditions for that. The temperature of the water. There shouldn't be any antibiotics or any disinfectant of any kind in the water because the vaccine virus might get inactivated because of that. So all these things need to be paid attention by the farm manager. Then vaccine handling, misreading of the label, giving it, not following the instructions, delaying the vaccination from the vaccination schedule, and as I mentioned earlier, removal of water sanitizers, and stress is also an important factor. So environmental extremes, so the temperature, overcrowding, that should be avoided. Nutrition or parasitical load, that also needs to be taken care of, and timing of the vaccination, like morning hours is supposed to be better, and if there's a heavy field load at that times, one shouldn't go for vaccination. So all these things need to be paid attention. And so the other point that I'd mentioned, especially with which is unique with commercial poultry, is the interference of maternal antibody. This plays an important role in vaccine failure because, as you said, this for animals is a vaccine. So, what is maternal antibody? So, it is the antibodies that are being transferred from the hen to its to the chicks through the egg. So and that's very important because that early protection from because field it's full of all kind of viruses and bacteria, and to protect the birds, they need to have that maternal antibodies in them, which defends the bird from these pathogens. But in case of vaccines, it has a kind of a negative role, you can say, because the those maternal antibodies cannot differentiate between the field virus and the vaccine virus. So they are going to neutralize both of them. So say these, normally whatever vaccination schedule is there, the IBD vaccine is given, which is a live vaccine, and as you can see, there are different flocks, and these different flocks will have different level of maternal antibody. So these are the different flocks having different level of maternal antibodies. So this greenish yellow line is the threshold line. So at this level of maternal antibody, the vaccine below this line, the antibody titers below this is where your vaccine will be able to replicate. And anything above this line, the antibody titer above this line, here the maternal antibody will neutralize the vaccine virus. Yeah. So it. So now the question arises. So now the question arises that for these different flocks, the optimum time of vaccination is different. So for flock one, it would be somewhere around say day 5, but for flock 2, it is around day 10. Flock 3, it is day 15, and for flock 4, it is around day 20. So then how do you decide that? So that's a major drawback, because for each and every flock, you cannot change your vaccination schedule. So that is a kind of a big issue in vaccination for infectious bursal disease. So I hope this point is clear. We'll get back to this point and how vaccination strategies can prevent this questionable timing of the vaccines. Um, the next point for vaccine failures, as I discussed, was vaccine challenge mismatch. So, say vaccine serotype, especially for adenoviruses. This every now and then the serotype which is circulating changes. So, and by serotype, you, as you understand, serotype of vaccine of one particular serotype cannot protect against a challenge virus of another serotype. So the serotype has to be matched. So that is why good surveillance is needed. Vaccine strain changes. Again, there are mutations going around with these viruses, or especially RNA viruses, and there are their antigenic epitopes, they change, and giving rise to variants. This is especially important in cases like infectious bronchitis. And so the vaccine also has to be upgraded accordingly to match the vaccine strain that is being circulating. And vaccine pathotypes, this is especially important in Newcastle disease virus. So in Newcastle disease, it is just one serotype. So technically, Lasota should protect against your all different pathotypes or your velogenic, mesogenic viruses. But it doesn't happen that way, because there are these point mutations which changes, and for getting a better protection, it's not that Lasota will not protect; it will cross protect against different pathotypes. But to get a better protection, you need to be as close antigenically as the field virus that is being circulating. So again, viruses need to be updated accordingly, and that's where technology comes in, where more reverse genetic vaccines are being used, or particular genes from circulating field viruses are taken and inserted into Lasota, and that way you have a safe vaccine, but still which is more relevant to the field challenge virus. And we'll get to this point later in the talk. Vaccine modifications. So cutting dose, that's quite a common issue in the field. Combining different vaccines, especially live. So certain vaccines can be combined, certain vaccines cannot be combined. So one needs to be aware of the do's and don'ts. And vaccination only for threats that are prevalent in the area. That is another issue. Cost cutting thing people do that say there are no much IBD outbreaks, so they skip IBD vaccination. And sometimes veterinarians also recommend that, but there are times when one shouldn't do that because you're not aware of the situation. Maybe not in your area, but in a close by area, there might be outbreaks of bursal disease. So one should consult the veterinarian before deciding on making such changes to cut vaccines or avoid vaccine. Uh, so area of focus for vaccine research is, as I mentioned, the new circulating strains. So one has to be aware of what is out there, and then accordingly uh, do cross protection studies. And there is no protection, then update the vaccine with matching field virus. And this is especially how here we are always at Bentley's, we are on our toes on this issue, and regularly we update our killed vaccines with whatever is the circulate to match whatever is in circulating field viruses, and introduce that as a kill vaccine. And also there are new serotypes and new pathotypes, so we keep looking for that and update our vaccine accordingly. There are new circulating viruses, so virus discovery is also an important tool. And with NGS and this whole genome sequencing tools available, virus discovery is much easier for discovering unknown viruses. Um, and once we have known and we are able to culture it, we can introduce that, try to attenuate it, and or give it as a kill vaccine, and also develop new vaccines, as I mentioned earlier, for old viruses. Based, this is more like a strategy-based vaccines or concept-based vaccine. So we'll touch more and discuss more on those later. So before we get more into the innovations in vaccines, I would like to just go back to basics of how exactly immune response is elicited. So here, as you can see, this is a dendritic cell or antigen presenting cell. This is CD4 T cell, and this is a CD8 T cell. So here you can see this is a virus which is engulfed by a dendritic cell. It's broken down into little pieces called peptides, and then these antigenic peptides are then presented by the class one, MHC class one and MHC class two, and as we all know, cells, while class 2 presents to CD4 T cells. And uh, once it's presented, then the appropriate T cell receptors of CD4 T cells and CD8 T cells, they get engaged, and the core receptors also get engaged, and that gives a signal for secretion of different cytokines, and these cytokines then drive an immune response by this proliferation of this T cells, and they migrate to the infected tissues, and if it's a CD8 T cells, it causes lysis of the infected cell. It just kills the infected cells. And if it's CD4 T cells, it will then disengage first with the dendritic cell, and then and engage with a B cell. And again, something similar what interaction happened at the junction of DC dendritic cells and the T cells. Something similar happens, but this time it's a B cell and a CD4 T cell, where it, based on its interaction earlier with MHC, it instructs the B cell of what kind of antibodies it is supposed to produce. And taking that signal, it senses uh, the the peptide and generates a repertoire of T cell receptors, which then give rise to antibodies. And then these antibodies can then, when there's a infection of a real virus, it can neutralize it very well. So, and once that is done, these B cells go into their population gets reduced, but there are few which still stick around for sometimes the duration is for few months, for some viruses or bacteria it is for lifetime or for several years. So those are called memory B cells. So these B cells then become memory B cells, and these are and whenever they come across the virus, they just go into multiplication phase and just generate mount a very strong immune response and bypassing this whole uh, instruction regime which we discussed earlier. So this takes time. So, but once it's already pre-exposed, the antigen has been exposed, so these memory B cells can act immediately and mount a very strong response. Within a couple of days, you get a very strong and that is basically the idea of vaccines, and that is how vaccines work. So, give a strong antigen which can give you a better CD8 T cell response, and within CD8 also there are memory CD8 T cells and these memory B cells. So the memory cells are what a good vaccine should be targeted. And so, as you can see, the most governing factor, which whether a vaccine is going to work or not, uh, is depends on what is going to happen over here in this interaction. So, where the peptide is being presented to the T cells, to the CD4 T cells. So that is very important, and it is important what kind of antigen is being given or being presented to the dendritic cells. So that antigen or the immunogen is nothing but the vaccine, and how best it is presented by to the dendritic cell so that the dendritic cell can in turn present it to the T cell. So that is basically how, in short, the immune response works for that. So, as I mentioned, this interaction is very crucial, and this peptide is what governs the immunity. So, so just to classify or to break it down, the vaccine research focus can be divided into choosing the right immunogen, adjuvanting it in the right way, uh, and not just adjuvanting it in the right way, but also delivering it in the most effective way. And also, the research focus is vaccines with some X factors. So, some diseases are unique in a certain way. So the vaccine has also needs to cater that uniqueness and so that it can protect against that particular pathogen. So choosing an immunogen. So while choosing the immunogen, traditionally most of the vaccines have gone for whole virus. So basically, this it's inactivated with formulation of the BPL or any of those inactivating agents. Any of those inactivating agents, plus exactly, especially this is important for mutating viruses like avian influenza virus. So within avian influenza, there is a matrix protein which is quite conserved, and since it is very important for particular function in its life cycle, so you don't notice any major mutations in the matrix, because if there is any mutation in the matrix gene, the protein is not going to be functional. So that is why it has been maintained as quite conserved. So this can be exploited as a way to choice as a choice of immunogen, as it doesn't vary, and any antibody against this particular protein would be able to give a uniform protection, irrespective of the mutation happening in the virus, or even protect different subtypes in case of influenza. However, there are some issues in using this matrix. There has been a lot of interest in making a universal vaccine. So, in that, matrix protein was considered very heavily. Um so, a universal vaccine would be this one vaccine would protect against all subtypes. As we know, there are so many subtypes within influenza. Like you have your H1, H5, H9. So it would be really nice to have just one vaccine which will cross protect against all. So that is the idea behind it. Next is the antigen eliciting neutralizing antibody response. So with IBD-VP2 vaccine, so that's a great example for in poultry. So the BP2 gene it governs the neutralizing epitopes. And so, if you have those in your vaccine, that is able to protect, and you don't need to have the whole virus. Just having the IBD-BP2 protein can protect against the bird against the virus. So there are a lot of advantages of this kind of vaccines, which we'll discuss. And there are also the peptide form of vaccines, as Dr. Kutre sir mentioned, that peptides are very important, and that's a new trend in designing ELISAs. So it's also uh, similarly quite important in for vaccines because these days synthesizing peptides is much easier and has become much cheaper. So instead of uh, uh, getting, so it's just coming down miniaturizing to a level where before the whole virus had to be used, then it's coming down to either the conserved protein or the antigen which elicits neutralizing antibody. Only that protein can be used, and even further down now it's only coming down to the peptide. So as I mentioned earlier, so in this graphical representation, what is being presented by the MHC is nothing but just the peptide. So the idea behind using peptide vaccines is if that is what is going to govern the immune response, then why give the entire virus when you can just provide just that little peptide to fill it. So there is a lot of experimentation going on in that way. Even vaccines because in as much only as extra. So for safer vaccines, that is the idea behind using peptide based vaccines. And as I mentioned, now coming down even below that is instead of using the peptide, you can come down to a level where you can take the genetic material which will make the peptide or make the protein in this case for neutralizing antigen. So the DNA that is required, or nowadays a lot of interest has been developed in using mRNA vaccines. So the mRNA, because from the holy grail from from DNA you get the RNA, and from RNA you get the protein. So giving just the mRNA, which will then directly get expressed in the target cell into the protein and get presented onto the antigen presenting cells and induce an immune response. So that is how the research is getting deeper and making it more specific, I would say. So, adjuvanting the vaccine, as we are all aware, a lot of common adjuvants have been used to give a better and stronger immune response. And within that, there's been a lot of development in the area of vaccine adjuvants, and still the traditionally used mineral salts like aluminum hydroxide is still commonly used, and we know a lot of them that are being used. Emulsions like MF59, Montanide, Fluence adjuvant. So these are also very commonly used and still very popularly used. Um, and in recent years, these kind of emulsion-based vaccines are not used only for killed vaccines, but they're also being used for live vaccines, for giving in ovo vaccines and for mucosal vaccines. So these Montanide gel and Montanide based vaccines can, these are commercial vaccines which are can be used for live vaccines as well. And there are also microorganism derived adjuvants such as cholera toxins, CPG, which is being used for DNA vaccines, and also PAMs, which is the pathogen associated molecular patterns. So these are basically in the virus, they are, or it is the innate immune system of the animal has over the years developed a way to induce an innate immune response looking for a pattern within different microorganisms. So, say, for example, RNA viruses have, especially at one of their replication stages, they make a double-stranded RNA. So these double-stranded RNA is common for influenza virus, for Newcastle disease virus, and for infectious bursal disease. So wherever that pattern is recognized, because in no case in animal within the animal's body is a double-stranded RNA prepared. So that it happens only in case of viruses. So uh, the innate immune system has developed a way to recognize that kind of a pattern of double-stranded RNA as a as a foreign invasion of a pathogen, and can immediately uh, start a chain reaction, if you will, or oh, what I'm looking for. Signaling pathway. That's, yeah, signaling pathways, which can then induce a very strong immune response. So poly-IC is something similarly. It is a double-stranded RNA. CPG is which is again very particularly noticed in cases of herpes viruses. So these patterns are recognized, and then an innate immune response is kicked in, which can control the disease. So these can be used as vaccine adjuvants. So that is how the adjuvants. So these kind of adjuvants can be used in for live as well as killed vaccines. And then you also have immune adjuvants. So as I mentioned earlier in the same graph, just go back real quick. So here, once this interaction happens between the antigen presenting cell and the T cell, there are these cytokines which send the population of the T cells. So the idea of using cytokines itself help for the T cells. This for the dendritic cell, they secrete cytokines such as TNF alpha or IL-6, which help in proliferation of these T cells. So the idea of using cytokines is kind of mimic this. So instead of, this interaction will happen, but if the interaction is not very strong, if you artificially provide IL-6 or TNF alpha for that matter, or IL-2 for that matter, the T cells will believe that will, or I won't say believe, but they have receptors, and these receptors will get occupied by these cytokines, and then put them in a mode of expansion, and they will start replicating. So you're kind of bypassing this interaction and giving them cytokines directly, so they can induce an expansion and mount a good immune response. So so that's the strategy of using these kind of cytokines. And these cytokines can be incorporated into live vaccines. So, say you have a vector based vaccine where you express the antigenic region, but along with antigenic region, you can also express, along with it, a particular cytokine, such as IL-2. And there are research articles where they've developed recombinant HVT vaccines to express IL-2. So these are interesting concepts in vaccine technology that can be used. Um, and so how adjuvants work, so as I already mentioned, so that is kind of summarized in this picture. But basically, there is this antigen presentation, and there is expansion of your T cells and B cells, and wherever there is an adjuvant, this expansion is much higher, not just for T cells, but also for your B cells and your plasma cells, giving a lot more antibody response compared to the vaccine which is given without an adjuvant. And different adjuvants, they work differently. So you might be aware of people talking about Th1 response, Th2 response. So for certain pathogens, the immune response that is needed is more Th1 like. For some others, it is more Th2 like. So accordingly, you can choose the adjuvant to match the pathogen of interest, and then decide which adjuvant you would want to go for. Then next, vaccine research is also being in the area of delivering a most effective way of delivering the antigen. Because if the delivery of antigen is not done in a proper way, vaccine would not be of much use. So in our textbooks also we have known that certain vaccines need to be administered via a certain route, and not certain routes. So, uh, as we understand, route of vaccination plays an important role. So is the delivery of the vaccine in even at a lower level, at a cellular level, is also very important. So there are virosomes which are used. So in virosome, it's basically, it mimics a virus which has the attachment proteins like a live virus, but it does not have any genetic material in it. So those are called as virosomes. Then there are microparticles which are used, or such as chitosan and alginate. So these can encapsulate antigen and protect them from the immune response. In this case, I'm talking about maternal antibody response, or pre-existing antibodies in case if the animal is already vaccinated earlier. So it can protect against getting neutralized, and also give help in slow release, and also in areas where you want to target it to the gut, but before going through it, your antigen has to pass through the gastric juices, which has very acidic pH, and at that acidic pH, your antigen will get destroyed. So such encapsulation would protect it in the from the from that low pH, and help it reach the target organ in the intestines. So, for example, like diseases like coccidiosis, or any gut-related pathogens. Then carbohydrates such as saponin, menan, and also chitosan, they uh, help in again kind of protecting the antigen, and also inducing multifunctional T cells. And there are then nanoparticles such as silica and gold nanoparticles, which stimulate strong innate immune response. And there is this dendrimer technology, one of the example is Starburst. Again, these are all more research level uh, vaccine delivery modules. There are not that I know of any real commercial products so far, but looking at the way the progress is being done, it's just a matter of time when these products will be out there in the market. So subunit vaccines and peptide-based vaccines, I'll just go a little more in detail of what we understand by that in real-life examples. So as I mentioned, IBD-BP2, it's a structural protein. So this is or IBD virus, and this is the protein IBD-BP2 being expressed. And here in pink, this is the genetic material. So for subunit vaccines or recombinant vaccine, what is done is this protein of interest is taken out and cloned into expression systems such as bacteria or yeast. And after induction, they produce this gene, this foreign gene, thinking as its own. It is produced and it is expressed, and it is let out into. It can be expressed either thrown out into the supernatant, or it can be part within the as inclusion body within the bacteria or the yeast. So it can be purified from it and then given directly as vaccine. So that's what we understand by subunit vaccines. That's how they are prepared. There are several advantages. There's rapid generation of vaccine once the methodology is established, because once because most one of the issues with the traditional vaccines is some of the newer vaccines, it takes time for it to adapt to certain culture systems, such as in to grow it in embryos or in cell culture. You need to do several passages, like hundreds of passages, and with that there are issues that it gets either too adapted to a different system, and then it does not replicate into the host when given as a vaccine. So it becomes either too mild or it's not enough attenuated, and then it can cause vaccine reactions and might cause a disease instead of protecting against the disease. So there is a rapid generation. Once you know that this is the positive agent and this is the antigenic region or the immunogenic region, you can just take that, clone it in a system, expression system, express it, and your vaccine is ready. So within, say, a month or so, you can have a vaccine right after an outbreak, which is ready. And this is what we are seeing in the corona outbreak also, that the first vaccine that was developed was the subunit vaccine, which was developed within almost 60 days, within 2 months of the outbreak. So and now one would ask, like, if it developed within 60 days, why it's taking so long? There are a lot of tests, safety and efficacy tests that needs to be done. So that's what is taking time. But the vaccine per se can be developed soon. And how effective it is going to be, that is a different question that needs to be studied. And that is what takes time. But if you have something that is effective, within a month or so, you can have a very good vaccine out there. So there is, so as I mentioned, you can tackle to a situation very quickly, and these vaccines are safe because you know these are already being tested. So like these systems where the protein is being expressed are already been tested for its safety, so there shouldn't be any issues with that. And the more effective than killed vaccines. This for quite a few years, this has been a drawback with subunit vaccine, that they're not very effective. But with these newer advances in adjuvant technology and in nanotechnology, like using nanoparticles or microcapsules, it is helping in making such subunit vaccines more effective. And these vaccinations can be done at hatchery level, so that way there's a uniformity in that. And it can also use DIVA strategy, because since these are recombinants, you can you can make changes, and you can identify based on the genetic or the kind of immune response that you're getting, and differentiate between whether it's a field virus or a vaccine virus. So giving you a really easy, quick example is talking about IBD VP2. So if you're making using a VP2 gene, a VP2 protein as a vaccine, so it's going to elicit antibodies only against VP2 gene, VP2 protein. But in case of a field vaccination, a field challenge, there it will also make antibodies against VP2, but it will also make antibodies against VP1 and other proteins of IBD. So to differentiate whether uh, the antibody response is against your vaccine, it will make only antibodies against VP2 and no antibodies against VP1. And if it's a field virus, it will make against both VP2 as well as VP1. So you'll have to design your ELISA in a such a way that you can differentiate between the two. So that's a very strong advantage. Drawbacks, there are some regulatory issues because these are not very common vaccines, um, and they can be expensive. And revaccination would be needed, but as I mentioned, with newer technology, people are working on that to make just give one shot, and it should give you a good immune response. And it needs skillful manpower because again, it is more like a kill vaccine. So, but again, technology can help us in that. So I think everyone must have heard a lot about nanoparticle and nanotechnology-based vaccines and all that. But basically, it's nothing really fancy about it. It is basically anything that can be measured within nanometers is called a nanoparticle. So it's as simple as that. And but it is a very good tool, and it can be exploited in lot many ways, more ways than we can imagine. So basically, nanoparticles are taken, and the antigen is taken, and then they are mixed. So here it gets adhered to the nanoparticle, the vaccine, either the antigen or the virus, and once inoculated, this nanoparticle itself can act as an adjuvant, as mentioned here. So, so there is activation and of cytokines such as IL-1 and TNF, and helping in maturing the dendritic cells and getting a good immune response. Also, nanoparticles, they can be used for targeting particular tissues. The other concept is encapsulation. So as I mentioned, like if you want to protect it from acidic pH of the gut and reach to the intestine, you can encapsulate it. So here is a good example. So here is the protein, which is basically the antigen or immunogen, and here it is coated with chitosan. And in this particular paper, they coated, so as you can see here, so it is encapsulated. The protein, the antigen is encapsulated by chitosan, and then it is cross-linked along with alginate, which is another you can say an encapsulating agent. So in this particular paper, they coated it with two layers. One was chitosan, and then along with chitosan, it was coated with alginate. And here is how it was being exploited. So this is getting into the gut. So having these two coatings, the alginate, it protected the antigen from breaking down in the acidic pH. If it was only chitosan, most likely in the acidic pH, it would have broken down, and your antigen would have just gotten inactivated. But because of this alginate, it can pass through the stomach, and once it reaches its target organ here in the intestine, here is the blow up of what happens. So, so this is your uh, the coating where now the alginate layer has come off, and here only you have the chitosan. Now the chitosan coated antigen is getting into the epithelial cells of the gut intestine, and then getting into the dendritic cells and as you can see here, and being presented to the T cells and inducing an immune response. So this kind of gives a schematic of how useful, how the encapsulation helps in more effective vaccination. Uh, Lastly, I would like to talk about the X factor. So, as I mentioned earlier, so with particular diseases, there are some unique things about a particular disease. So, to cater that issue, some vaccines have to take that into consideration. So, in especially in poultry, we want a vaccine strategy which is uniform and is easy to do. So hatchery vaccination is very common. In gene gun, it is not practical for poultry, but in other species it can be done. And so this is for inactivated vaccines, and for live vaccines, have a strategy of multivalent vaccines. We'll get little more in detail on multivalent vaccines. That is where vector vaccines come in. So when you talk about multivalent vaccines, I'm talking about making easy vaccinations. So say in poultry, we vaccinate routinely for Newcastle disease virus. These are more important disease like Newcastle, bronchitis, infectious bird cell disease. These are routine vaccinations. So if we can make a multivalent vaccine using a vector-based vaccine, where that one vector, when vaccine, has the antigenic region of IBD as well as Newcastle disease virus, maybe as well as infectious bronchitis, all these are packaged into one because as I mentioned, what is really required is just the antigenic part. You don't need the entire virus. So only the relevant part of, say, Newcastle disease virus and IBD virus and say bronchitis virus, if these are just cut out and pasted into a vector vaccine, which is replicating. So you have a live vaccine which is going to replicate, and it will replicate the antigenic portions of the more important viruses. So that way, just giving one vaccine can take care of 3 or 4 diseases at one time. So we'll discuss this a little bit more in detail. And as I mentioned earlier, DIVA strategy, so to differentiate between vaccinated and infected animals. And the other important area of catering is pre-existing antibody, such as in that again encapsulation can be useful, but also a very novel idea is on using immune complex vaccine, and we'll briefly discuss the two things. So immune complex vaccine is basically where you're just mixing the vaccine strain along with the specific antibody at a particular ratio, and that way it hides the virus and kind of from the antibody from the maternal antibody. When this immune complex vaccine is given into the animal, it gets protected because the maternal antibody cannot bind to the structural protein of the virus because it's already blocked with the this complex. So how this vaccine works is okay. I'll just like to go back and just for this one slide, switch to this one. So just to understand how immune complex vaccine works. So as I mentioned earlier, so there's this maternal antibody which drops with the age of the bird. So early on, there's a lot of higher maternal antibody, and with time it goes down. And one of the issues which we discussed earlier was how do we know when to vaccinate? So because if the maternal antibody is high enough, your vaccine virus is just going to get neutralized and before it can induce an immune response. So, so the immune complex can hide from the maternal antibody. So this is how it would work. So here, if say on day 0 itself, you give the immune complex vaccine. This is the immune complex vaccine. This is the virus, a live virus, and the blue is the antibodies bind complexed with it. So with time, this immune complex is going to release the virus out. And this happens in proportion to the drop of the maternal antibody. So as the maternal antibody is going down, the the complex is going to get broken down, and the virus vaccine is going to get released. And now this is the the right time when this vaccine virus is released. That is the time when the maternal antibody is released to such a level that is below the threshold, and this vaccine virus cannot be blocked by the maternal antibody because the antibodies have gone down, and now that gives the chance for this vaccine virus to multiply, and then it replicates, and this replicating virus can then induce a strong antibodies. And also, when this there is there could be a possibility that there is a window of susceptibility, because in this stage you don't have antibodies, because this is when the virus is developing the antibody, so it takes some time. It just cannot happen in immediately. So this can be a possible window of susceptibility. But the beauty is with immune complex vaccine is the since this is also IBD virus, it occupies the same tissue within the animal. So the vaccine virus also goes into the bursa, and the field virus also goes into the bursa. But the advantage this virus has is it is already there. So it beats the the field virus by occupying the site, and so it gets occupied the target organ, and the field virus cannot go there. Even if the antibody is not developed, the target organ is already occupied by the vaccine virus. So it's, in a way, you can call it as a competitive inhibition for the field virus, and that is why the field virus cannot attach to the target cells. And and after that, antibodies are there. Once antibodies are there, it doesn't matter. The antibodies are going to protect against the field virus. So that is how the very interesting part of immune complex, how it works. And I just thought it's important that I should take the time to explain it. Okay. So again, there are a lot of advantages. Mainly, you don't have to wait for the maternal antibody, or there is no guesswork involved. You can just vaccinate at day of age and get a good, long-lasting immune response. And also, it can be given at hatchery level, so there is uniform vaccination. Drawback, one of the biggest drawback with this vaccine is it is cost intensive, because you actually, to making the complex, you really have to make the take the serum and mix it with the virus, and that has to be at the right ratio. So it's a very expensive vaccine. But looking at the advantages, it's most it's a cost benefit analysis one needs to do, and go for it. Lastly, I will talk about vector vaccines. As I mentioned, this is also very I won't call it a new technology. It's been around there for almost more than a decade or 2 decades. But in India, it's a newer concept where, as I mentioned, Marek's disease vaccine, HVT, herpes virus of Turkey, is used as a vector. And in this vector, is in this example, this is a ND virus. So the F protein, which is the antigenic gene, the fusion protein, which is expressed on the surface. So the gene that expresses that protein is taken, and it is cut out from the ND virus, and put into an appropriate region in the HVT genome in such a way that the HVT virus, when it replicates, it will consider this F protein of ND as itself, as its own protein, its own gene, and express the F protein of NDV along with its own other proteins. And since this is antigenic protein, once this protein is expressed in the chicken, it will induce an immune response. And as we talked about multivalent vaccines, so instead of taking just the F gene here, you can also put multiple genes from, say, VP2 of IBD and the S1 gene of bronchitis. You can insert multiple antigenic genes from different viruses and put them into these HVT as a vector. And just this one vaccine can cross protect you against multiple diseases. So this is a very innovative way of and very easy and sure shot vaccination way of vaccination. And since it can take care of multiple vaccines in one, it can be more cost effective in that way. So again, in this maternal antibody does not matter. Say in case if you put VP2 region of IBD, because the response that HVT generates is more cell mediated, and it's a cell associated virus. So it transmits from one cell into another cell. It doesn't come into outside the cell. So there is no chance for the maternal antibodies to bind the vaccine to the HVT and neutralize it. So that's a very strong advantage. There is a quick response, as I mentioned. Say if there's a novel virus you don't know much about and it's not growing well in other systems, you can just take that gene and insert it into the HVT vector and make a vaccine which is specific not just against Marek's disease but also against the new disease that you have encountered. There is persistent expression since this is herpes virus. It's a persistent infection. So since it's persistent, its replication, the protein is also expressed throughout the life. So you don't need to boost it with anything, giving a very long-lasting immunity. And again, here also, this is as we all know, Marek's disease vaccines can be given at hatchery level, so that's the other advantage, and it can be used as a DIVA strategy. Again, the drawbacks, since it's HVT, cold chain has to be maintained because, as you know, HVT vaccine has to be cell-associated vaccine, so it has to be kept in liquid nitrogen. So that's one of the drawbacks, and it can be expensive, and especially for broilers, and you need skillful manpower for the vaccine. Vector cassette vaccine, this is again similarly to that what I discussed. Reverse genetics, this is, I won't go too much in detail because I guess I'm running out of time. So I won't discuss this in much detail. But basically it's the 8 segments. So influenza has 8 segments. So all these 8 segments are cloned into plasmids and then transfected into a cell. And you can get a virus out of it. So the beauty of this is from these 8 plasmids where you have expressed these 8 different genes, you can do any manipulation you want, and you can cater to it. So say you can use this for even for uh, Newcastle disease, but in influenza, say you have a vaccine strain and you have a new outbreak of a new subtype, new H5. So you can take H5 of a circulating virus and put it in this plasmid and incorporate all other genes from your vaccine strains and make a reassorted virus which is more relevant to your region. So that's the idea behind it. Towards the to end this, I would just like to say that all this vaccine research is being driven, and we are in an era of package. We all like to hear this word and have it in a package form. So just presented in such a way that you just do this one thing and everything is taken care of. So you give this one vaccine and you don't need to give multiple times. So if you give one immune complex vaccine, you don't have to vaccinate it or give booster or anything. That one vaccine is good enough. So like multivalent vaccine, if you give one recombinant HVT vaccine, you don't need to vaccinate for Newcastle disease, infectious bursal disease, bronchitis. So that is where this research is going on, and just in a way reducing cost, reducing effort, reducing manpower, and at the same time having a very much more highly effective vaccine. So clone vaccines are also part of it. So and as always, you cannot have a single strategy that fits all and in all geographies. So that is the other aim of having a single strategy which can fit in all geographies in all areas. Yeah, that's about it. And yes, has its uniqueness. Every region has its uniqueness, like manpower, biology of viruses. Viruses keep changing, so that is also being taken care of. So, just to summarize, discuss the major change challenges. Diagnosis is very important, not just at molecular level, but also serological level, and finding solutions and improving current vaccines for today's competitive world. Not just on using knowledge of viruses or the host or the immunology of the host, but also the new available biotechnological tools and how they can be applied in vaccine research. So that's also very important. And knowledge and understanding of the disease, which is age old, and that no one can change that. So always our old strategies, our old way of understanding the disease. There is no option, no alternative to that. So always that is needed. So with that, I'd like to end, and I'm sorry if I've taken a lot more time, I guess. But thanks.